Alright, welcome to my presentation about DNA technology. So first we're going to talk about transgenic organisms and plants. A transgenic organism is when foreign DNA is introduced via biotechnology. Foreign DNA, or the transgene, is described as DNA from a different species of recombinant DNA from the same species that has been modified in the lab and reintroduced. Though this image of taking out a section of DNA may not look significant, this section of DNA may be from the same species that's been taken out into the lab, modified, and then reintroduced to the animal, therefore creating a transgenic organism. It can also be a section of DNA that's from a different species or organism that's introduced there without DNA modification from the lab. For DNA sequencing and PCR, DNA sequencing is the method of determining the nucleic acid sequence in order of nucleotides in DNA. This involves any system or technology for determining the order of the four bases. So for the sake of the PowerPoint, A, G, C, and T. Now, PCR is a polymerase chain reaction, and it is a widely used technique for rapidly generating millions to billions of copies of a specific DNA sample, which allows scientists to take a small sample of DNA and amplifies it large enough to study it in detail. Here in these images, we see a sample of the DNA, and we can see the bases in it, the A, C, T, and G, which was mentioned before, and then an example of the polymerase chain reaction. So we see the second cycle, the third cycle, and then at the end we see our PCR outcome. And at the beginning we see it get separated and go from a DNA sequence all the way into a PCR. So now to talk about DNA profiling. So DNA profiling is a method of getting a specific DNA pattern, which is referred to as a profile, from a person or a sample of body tissue. Even though we are all unique, the majority of our DNA is identical to that of other people. Specific locations of the DNA in each person is different, and that causes a great difference in people, which is why we're all different, even if we're so similar. Here we see a DNA profiling test, which is actually, if you look over here at the left-hand side under the KB, we see a 2-3, a 4-3, a 6-5, a 9-4, a 23, and a 1. So on these areas, you can clearly see it. There's a specifically dark spot. Now, this is significant because this is how they can use DNA profiling to determine genetic capability. So basically, to determine which of these letters up on the right would be matching to this person, we would figure out who has the most matching to the highlighted areas on the left. Now for stem cells are cells that have the ability to differentiate into a number of different types of cells in the body, which we're going to talk about. And because they're unspecialized, they're unable to perform certain bodily activities. They have the capability to differentiate into specialized cells, which we're going to go over in just one second. So here we see those cells. So we have sex cells, muscle cells, fat cells, bone cells, blood cells, nervous cells, epithelial cells, and immune cells, which all generate and stem from stem cells. This is why stem cells are so important to the body, because they have so many functions and they help us with so many things. And without them, we wouldn't have any of these here cells that we need. So on to cloning. Cloning is a process that allows scientists to create perfect genetic duplicates of a living organism. Cloning can be done on genes, cells, organs, and animals. Identical twins are comparable to clones because they have identical genes. However, it's not exactly the same thing. So hopefully we've all heard of the whole goat that got cloned and everything, which is a really difficult process, which is why it was a big deal, even though, to me, I thought, they all look pretty similar, but there is a big difference. They were genetically identical. Here you can see an example of how they create an identically genetical clone. So DNA used in medicine is very important. DNA testing can be used to diagnose inherited disorders, investigate malignant processes, perform forensic medicine, and discover infectious organisms. After the introduction of DNA analysis, disease and diagnosis has vastly improved. And here are all my sources cited. They're listed by slide and topic. So I have topics on slides 2, 4, 6, 8, and 12, including 10. So I hope you have a good day, and I hope this was interesting.